pelvis is one of the most sexually dimorphic bony elements of the human body. It is widely used in the forensic sciences field to estimate sex of recovered remains. However, there are several limitations when we use the pelvis. The first is the pelvis isn't always recovered at an excavation site. And if it is recovered, it is usually highly fragmented, which means that sex estimation methodologies that require an intact pelvis cannot be used. What if there's a probability that another bony element can replace the pelvis or complement the pelvis when we estimate the sex of recovered remains? For example, the crania. What if there's a probability that the crania can do the exact same thing that the pelvis can do? My study focused on assessing 22 unique interlandmark distances that were constructed using 23 cranial landmarks from 3 cranial regions: the anterosuperior, anterolateral, and posterolateral cranial regions. 22 skulls, 11 male and 11 female, all of Southeast Asian origin, were used in this study. The main part of the study was to conduct a few data analysis to assess the existence of sexual dimorphism in each individual interlandmark distances, rank these ILDs, interlandmark distances, from the most sexually dimorphic to the least sexually dimorphic, and the most important part was to produce discriminant function equations that can be used to classify sex using portions of the skull. The study found that 8 out of 22 of the ILDs had a significant p-value, and the anterosuperior cranial region showed the highest sexual dimorphism compared to the other 2 cranial regions studied. The ILD jugale to glabella showed the highest sexual dimorphism with an original and cross-validation accuracy of 86.4%. 5 discriminant function equations were selected, ensuring that the original accuracies and cross-validation accuracies of the equations are the same. The accuracies of these equations ranged from somewhat 72.7 to 86.4%, and 3 of them were from the anterosuperior cranial region. In conclusion, the anterosuperior cranial region was found to exhibit high levels of sexual dimorphism and can potentially be used when the pelvis is not present at the excavation site. However, as this study was only a pilot study assessing the use of several cranial regions and ILDs, a small sample size of skulls consisting of only one geographical group was used. Therefore, this study needs further validation using a larger sample size of skulls consisting of more than one geographical group. Nonetheless, this study has proven the potential use of the crania, especially the anterosuperior cranial region, in estimating sex.